Okay. Good morning or good evening. Sorry, good evening, dear doctors. Hope you are doing well. Of course, as I show in the screen, focus on your goals, not on the obstacles, and that's the most important key to success. So today we are going for the second class. In the beginning, I would like to say that, see, I heard that in between the first class, in our first class, there was some internet connection problems. So I'll be taking it later, maybe in the next class. The few topics that I was not able to convey to you properly is mainly on heme catabolism. That we will take later. Today, I'll be dealing with the topic of protein and amino acid, from their protein chemistry and amino acid metabolism first. Okay, we'll straight away go to the subject. First, on the chemistry of protein. A protamine, peptones, prolamine, lactalbumin. Which of the following is derived protein? So we know that proteins are made up of amino acid, and protein, there are mainly three types of protein. One we call it as simple protein, and other one is conjugated protein, and third is derived. If the simple protein or conjugated protein on partial hydrolysis produce some derived protein. So peptones, the peptides, they are produced by the partial hydrolysis of the protein. So they are called as derived protein. We'll have some ideas about the simple protein to say about some importance of protamines and all. Okay. Conjugated protein means if a non-protein part is attached to the protein, that is called as conjugated protein. An example is our lactalbumin. Okay. What about simple protein? There, the first type of simple protein is albumin, soluble in water, but is coagulated on heating. Globulin, insoluble in water, soluble in dilute salt solutions. Now, for the third word, protamine. That's also a simple protein. It's a protein that is soluble in water, dilute acid and alkali, rich in arginine and lysine. What is the clinical importance of it? See, protamine zinc insulinate is one combination what we give for the insulin injection. It can slowly give out the insulin. So for the intermediate functioning, that means not fast, not slow. Intermediate, it acts by intermediate method. So in the sense, not so fast and not so slow. So that's a combination. Protamine zinc insulinate is one clinically important compound where protamine is used. And also, very important might be in surgical cases, in cardiology cases, where we give the heparin. Some anticoagulant effect of the heparin can be reversed by giving the protamines. That's also another importance of protamine. Then comes prolamine, the gliadin, gene of corn, gliadin what is in wheat. So they all contain the prolamine. They are rich in proline, soluble in around 70 to 80% alcohol. And lectin is another one, which has a high affinity for sugar. So whenever you want to separate out the carbohydrate containing compound, we use the lectin. So these are some important simple protein. And one more protein, what is soluble in strong acid alone, scleroprotein. So just not about this term, protamine, prolamine, and lectin. Albumin, globally, you are familiar through the plasma proteins. Okay. Next is about the structure of protein. So I just tried to say the classification of protein. Now to the structure of protein. Tertiary structure of protein is maintained by all except. And here, as you should understand that secondary, tertiary, and quaternary structure of protein. Hope you know that the protein have the structure at 4 levels. Primary structure, we explain the structure of protein for 4 levels. Primary structure, secondary, tertiary, and quaternary. In that, primary alone is maintained by covalent bond, mainly peptide bond and some disulfide bonds. Secondary, tertiary, quaternary is maintained by non-covalent bonds. Okay, so that whenever you heat, whenever some chemical, when it is acted upon by some chemicals, that structure, secondary, tertiary, and quaternary structure get destructed. On denaturation, only primary structure exists. So in this, hydrogen bond, hydrophilic bond, and ionic bonds are non-covalent bonds. So here the question is, tertiary structure of protein is maintained by all except. So here is all about the disulfide bond. Disulfide bond, so the peptide bond is the one that helps in the covalent bond mainly. That covalent bond is the one that maintains the primary structure. All other comes under non-covalent bonds. Okay, just about the structures, primary, secondary, tertiary, and quaternary structure. What is the primary structure? Deals with it's a sequence of amino acids. Number and sequence of amino acid we call it as primary structure. And then what is the secondary structure? Secondary structure means actually local conformation. When we see this one, tertiary structure, when you check the one side, how it looks like, that is called as that structure, what we see in locally, we call it as secondary structure. It can be either alpha helix or beta pleated structures. Okay, so secondary structure means local conformation, and tertiary means a three-dimensional structure of one polypeptide chain. And quaternary means if a protein contains more than one polypeptide chain, then they exhibit quaternary structure. Okay, so hemoglobin, example is hemoglobin. It contains two alpha chain and two beta chain. Two alpha chain and two beta chain. And then it has two major quaternary structure. One we call it as T form, other one R form. For hemoglobin, there are two quaternary structure, T form and R form. T means tense. That means there a lot of hydrogen bonds or non-covalent bonds are there between these chains, alpha and beta chains. So its oxygen affinity will be very less. That is exhibited by deoxyhemoglobin. And the oxyhemoglobin will be in R state, relaxed state. Okay, so these are the four major structures, and only the primary structure is maintained by covalent bond. All other bonds are maintained by non-covalent bonds. And this is the school of thought I will try to tell you. Earlier, disulfide bond also was considered as a covalent, and that is a part of primary structure. But now, in all the books, comes under the fact that that also is considered to maintain the secondary structures or tertiary structure. So that is also is brought under the non-covalent bond. Okay, that's why here the answer is none of the above. Next, about the chemistry of amino acid. Indole ring is present in. We are supposed to know some structural details of the amino acids. And here, the indole ring is in tryptophan. Now, just we'll try to say some of the idea, some structural features of each. Here in this, all four options, tryptophan is the answer. And in all four options, about histidine, you should know. Histidine contain, please note, histidine contain a group called as imidazole group. And what's the importance of that group? That has the maximum buffering capacity. Because the histidine, this imidazole group's pKa value is near to our physiological pH. So histidine has the maximum buffering capacity because of imidazole group. So please note, everybody, please take down that word, imidazole group of histidine. And that histidine has the maximum buffering capacity. Now I'll just say about some chemistry about the amino acids. I try to classify it. One classification is aliphatic, aromatic, basic, acidic, And last one, imino. Which is amino acid? Proline. Not amino, it's imino. Because that amino group is there in a ring, not separately. In all other amino acids, amino group is separately standing. NH2 will be there. But here, NH will be attached. It will be the part of a ring. So, proline is an amino acid. Okay. We'll say some example. Then, in the aliphatic, we classify it into branch chain amino acid. We should know that. Then another type of classification is essential and non-essential amino acid. What do you put essential amino acid? Amino acid that cannot be inside the body, that should be supplied to the diet. There are 8 essential amino acid, 2 semi-essential amino acid. All other 10 are non-essential amino acids. And one more, amino acid that contain, protein that contain all the essential amino acids called as complete protein. Our egg albumin, milk protein, casein, example for complete protein. And if it lacks one essential amino acid, that protein is called as incomplete protein. And if any protein lacks more than two or more essential amino acid, if it lacks 2 or more essential amino acid, they are called as poor protein. Okay, another classification, glucogenic and ketogenic. There comes again one specific example. Leucine and lysine are ketogenic. That means what? From that to leucine, lysine, you cannot synthesize glucose. From all other things, all other 18 amino acids, you can synthesize glucose. So they are glucogenic. These 2 alone are ketogenic. In that itself, purely ketogenic is leucine. Because lysine, what happened? If you combine with some other compound during the catabolism, and then it breaks, that some area can be utilized can enter the glucose pathway. So we say but leucine is purely ketogenic. Okay. So again, some other classifications, simple amino acid, glycine and alanine. Branch chain amino acid, valine, isoleucine, leucine. What's the importance of that? Metabolic disorder of this branch chain amino acid will lead to a maple syrup urine disease. The urine will be having a smell of charred sugar, and we call it as MSUD. A branch chain amino acid. Third is hydroxy amino acid, threonine, serine. Sulfur containing cysteine and 2-cysteine combined together form cysteine and then methionine. Fourth group is with amide group, asparagine, glutamine. Then monoamino dicarboxylic acid, aspartic acid, glutamic acid. They are the acidic amino acid. And basic amino acid, lysine, arginine are the basic amino acid. Diamino monocarboxylic acid. That means contain 2 amino group and then 1 carboxylic acid group. So they are basic also. Okay. Then comes aromatic amino acid, phenylalanine, tyrosine, tryptophan. So phenylalanine, tyrosine will be containing a benzene ring. Tyrosine will be containing again benzene plus amino group, phenol group. Tryptophan, as I said, indole group. Heterocyclic tryptophan, again, it's an aromatic at the same time, heterocyclic in the sense, one cyclic structure contained other than the carbon atom. So tryptophan and histidine are heterocyclic amino acids. And amino acid, proline. There are some derived amino acids. For example, hydroxyproline, hydroxylysine, gamma carboxylated glutamic acid, acetylated or methylated amino acid in histones. They are called as some derived amino acid that can see in protein. But there are some derived amino acids that cannot be seen in the protein. Ornithine, citrulline, homocysteine are some amino acid that cannot be seen, derived amino acid that cannot be seen in a protein. So far I was telling about amino acid, generally they are alpha amino acid. At the alpha position, there will be an amino group. But there are some amino acid without an amino group at the different position, not in the alpha position. Gamma amino butyric acid, beta alanine are some examples. Okay. So these are the major 20. Traditionally, we say 20. But later on, nowadays, we have identified some more amino acid. That's it. Okay. In that, I'll say this one. Leucine, lysine are purely ketogenic. tyrosine, tryptophan, phenylalanine, isoleucine are both glucogenic and ketogenic, and remaining amino acids are purely glucogenic. So please note these four are aromatic. Phenylalanine, tyrosine, tryptophan plus isoleucine is a branch in amino acid. These four are both glucogenic and ketogenic. Purely ketogenic, leucine, lysine. Okay. Now comes, we said this word, essential, semi-essential, and this term. Traditionally, we know there are 20. Now we have two more amino acids that has been identified. 21st is selenocysteine. Cysteine is an amino acid that contains sulfur group. And instead of cysteine, some contain selenium. So they're called as selenocysteine. That's the 21st amino acid. And the 22nd amino acid is pyrrolysine. Okay, as I told you, purely ketogenic is leucine. Please note that. Please make a note on it. Purely ketogenic is leucine. Derivatives of amino acid that are seen in protein. As I told you earlier, what are the hydroxy amino acids? These were some questions actually earlier that came. Hydroxyproline, hydroxylysine, gamma carboxyglutamic acid. Then earlier I told you, I think twice. Last class also I said that amino acid with maximum buffering capacity is histidine. Why? Because the pK value is very near to that of a physiological pH. Okay. Next question. Glycine is present in, or what are the major compounds produced from glycine? That's a question. Hemoglobin, heme, for its synthesis, glycine plus succinyl-CoA giving ALA. That's the first step in heme synthesis. Glutathione is gamma glutamyl cystinyl glycine. So it contains glycine. Purine synthesis also C4, C5 and N7. That means carbon atom at the positions of 4, 7 and 8 and nitrogen atom at the 8 positions are provided by glycine. Creatine also, glycine plus arginine giving guanostate plus methionine is creatine. So these all four compounds contain glycine or glycine. Glycine, these are the compounds produced from glycine. So all the above is the answer. Now, if that's a question, you should know some biologically important compounds produced from different amino acids. So we said about glycine now. As I said, heme, glutathione, purine, creatine. Other than that, glycine is a neurotransmitter. It helps in the detoxification process, conjugation of bile acid. These are something you should know about glycine. And then conjugation with bile acid. Name for bile acid, cholic acid or chenodeoxycholic acid. Second, about phenylalanine tyrosine. What are the major compounds produced, biologically important compounds produced from the phenylalanine tyrosine? Our pigment, the skin, melanin, then thyroid, thyroid hormone, then catecholamines are the three major compounds. Major compounds are catecholamine in the sense dopamine, adrenaline, and noradrenaline. Okay. Then melanin, thyroid hormone are the major compounds produced from phenylalanine and tyrosine. Because the first step, phenylalanine, first by hydroxylation converted to tyrosine. Phenylalanine is essential, tyrosine is not essential. Please note that. Melanin, thyroid hormones, catecholamine. And one more we can see about tyramine is a neurotransmitter, tyramine, by decarboxylation reaction. Okay, third, about tryptophan. What are the major compounds produced from tryptophan? We started from there, tryptophan contains indole group. Okay, the compounds are mainly a neurotransmitter. You know that word? Serotonin. Then melatonin, that determines our diurnal variations. Then comes a vitamin, niacin. These are three major biologically important compounds produced from tryptophan. Along with that, it can give formyl group, one carbon group that can involve in purine pyrimidine synthesis. So we can say the word also one carbon group is another thing about tryptophan. Hope you understood this thing. These are the major compounds produced from this thing. And one more you can say from glutamic acid, GABA. Our inhibitor neurotransmitter GABA is produced from glutamic acid. And then nitric oxide from arginine. Other two biologically important compounds. Two questions on urea cycle. Source of nitrogen urea cycle. And for sense of urea, how many moles of ATPs are utilized? We'll just go through now and then come back to this question. Okay, leave that. This is what? Urea cycle. First, site mainly liver, partially mitochondrial, partially cytoplasmic. What's the use of urea cycle? If you say urea synthesis, fine. But why urea synthesis? To detoxify ammonia. Ammonia from where is produced? During the amino acid catabolism, amino group is removed as ammonia. And during purine pyrimidine catabolism also, they also contain some amino group that is removed as ammonia. Bacterial metabolism produce some amino group. And that also produce some ammonia. So these all ammonia are neurotoxic. So these neurotoxic compounds are converted to urea. So that's the use of urea cycle to detoxify ammonia. Site liver, in the liver's hepatocyte, partially mitochondrial, partially cytoplasm. Source of ammonia, so you know, and then how the ammonia is transported from the brain is not from the brain. Ammonia is transported as glutamine. Glutamine gets ammonia and becomes glutamine. So we say first line of defense on ammonia toxicity is conversion to glutamine. Okay, please note that. When I say transport of ammonia, when the main one from the brain is glutamine, meaning when the ammonia is produced, that combined with glutamic acid and form glutamate. So to reduce the toxicity, to inhibit the toxic property on the brain, the first thing what happens is ammonia will be converted to glutamine. Okay, glutamine. So the first line of defense against ammonia toxicity. And then from the muscle, ammonia is transported as alanine. It will combine with pyruvic acid and convert to alanine. Okay. When you say about the energetics, we say how it is related to a TCA cycle. I just say about that. Then what's the importance of urea? Urea value is, for example, 20 to 40 milligram per deciliter in the blood is a urea level. And that level will elevates in kidney diseases, renal diseases. So urea level denotes the kidney function. If it is elevated, kidney dysfunction. Not only urea, both urea and creatinine. We call it as 2-NPN, non-protein nitrogenous compound. They contain nitrogen, but they are not protein. NPN, non-protein nitrogenous compound that shows about the kidney function. You can say it is urea and creatinine. This is about the urea cycle. The first step, carbon, just go through this. Carbon dioxide plus ammonia. So urea is NH2CO NH2. So one NH2 is from ammonia. Okay. To form carbamoyl phosphate. Then carbamoyl phosphate combined with ornithine from citrulline. Then citrulline enters into the first step, this two steps is mitochondria, then enters into the cytoplasm. Sorry, the first two steps in cytoplasm, then enters the mitochondria. Then citrulline combines with aspartate and form arginosuccinate. So second amino group is from aspartate. Arginosuccinate then breaks to form fumarate and arginine. Arginine breaks to ornithine and produce urea. So in the urea, two amino groups are given by by ammonia and other by aspartate. Now the second question is about the energetics. In this pathway, see, first step 2 ATPs are utilized, convert to 2 ATP. So 2 ATPs are utilized. Second, ATP convert to AMP is equivalent to conversion of 2 molecules of ATP into ADP. Okay. So again, that means here in this step also, 2 phosphate bonds are utilized. Here also 2 phosphate bonds are utilized. So totally 4 are utilized. But the point, this fumarate enters into the TCSI. Okay. And fumarate will convert to malate, malate to oxalacetate. At that time, one NADH is produced. From one NADH, you can synthesize, listen, you can synthesize 2.5 ATP. Then that oxalacetate converts back to aspartate. I'll just show that. I think it is not there. This is what all about. Our aspartate combines with citrulline form fumarate, arganosuccinate. Arganosuccinate release the fumarate. So this fumarate enters into TCS cycle. Fumarate will convert to malate. Malate will convert to oxaloacetate. During this reaction, you know what happened? One NAD plus is converted to NADH. NADH plus H plus. So from NADH, you get 2.5 ATPs are produced. This is plus. These two are, the first, this one, this two, so here minus 2, minus 2. 2 ATPs are utilized. But here you get 2.5. And then what happened to oxaloacetate? Oxaloacetate converts back to aspartate. So this is the relation between urea cycle and TCS cycle. Urea cycle, TCS cycle. So in the urea cycle, 4 ATPs are utilized. Here 2 and here 2. So 4 ATPs are utilized. That's why I put the minus. But here 2.5 ATPs produced. So 4 minus 2.5, 1.5 ATP is the net utilization is 1.5 ATP. So for urea cycle, you need 1.5 ATP. That was a question here. So 1 mole of urea requires how many ATP, how many moles of ATPs? For urea synthesis, even though you need 4, 2.5 you'll get back, so remaining 1.5. The ammonia group is provided by, or the nitrogen is provided by aspartate and ammonia. Hope is clear about urea cycle. Now come to the next question. In physiological system, nitric oxide is known to act through. Nitric oxide was known as the molecule of the decade of 2000 actually, in fact. 90s to 2000. At that time, it was identified. It is produced in the body through our arginine. With the help of enzyme nitric oxide synthase, you get nitric oxide. Okay. And this nitric oxide is actually not so, half-life is very less, converts to peroxynitrate and nitrite or nitrate. And then, what's the function of nitric oxide? Through cyclic GMP, it helps in the relaxation of muscles. That's the point. Nitric oxide acts as a smooth muscle relaxer. For that, you need cyclic GMP. That's the point. So that's the function through which it helps a number of things. In angina pectoris, at that time we give it triacylglyceride from which nitric oxide is produced. Okay, and our Viagra contain a chemical, sildenafil citrate, that will inhibit. Usually what happens? The cyclic GMP after functions convert to GMP with the help of enzyme phosphodiesterase. This phosphodiesterase is inhibited by sildenafil citrate or Viagra. So vasodilatation of corpus cavernosum will become erect. So erectile dysfunction is treated with Viagra through this only, nitric oxide. That's one function. Angina pectoris, vasodilatation occur. We give it as a drug, as a sublingual drug. That one will help with the dilation of the vessels. Again, through this way. Nitric oxide is the one which can relax a smooth muscle. Next question. So we said about the amino acid breakdown, then by urea we said. Major compound produced from amino acid we said. Now we'll try to say some disorders related to the amino acid metabolism. First one is about the carrier. Amino acid transport across the membrane, we need some carriers. Out of this, which one leads actually due to a defect in the amino acid carrier? As I said earlier, the maple syrup urine disease is related to branch chain amino acid. Branch chain amino acid defect in the decarboxylation. Please, I'll say about that later. Please, now you just note this one, decarboxylation. In fact, the process is oxidative decarboxylation process. If that is defect, leads to maple syrup urine disease. Alkaptonuria is a deficiency of an enzyme, homogenesic acid oxidase. It comes from the tyrosine metabolism. Phenylketonuria, phenylalanine hydroxylase. And cystinuria is the one that means presence of cystine in urine. Cystine in urine. Because cysteine molecules are there. Its transport mechanism is under effect. So cystinuria is the answer. I'll come to that now. This is a common thing. Some disorders related to the protein, like amino acid metabolism. First one related to glycine. We have two types of oxalates in urine. Hyperoxaluria type 1, hyperoxaluria type 2. Related to glycine metabolism. Amino acid, the enzyme defect is alanine glyoxylate amino transferase for the type 1 and type 2 glyoxylate reductase. Then comes the word homocystinuria. It is in the amino acid metabolism. The enzyme name cystathionine beta synthase deficiency. And third, as I said, maple syrup urine disease for the branch chain amino acid. The enzyme name branch chain alpha keto acid dehydrogenase. It is an enzyme that helps in the process of, as I said earlier, oxidative decarboxylation. Now, two words, cystinosis, cystinuria. Cystinosis, cystinuria. Cystinosis, when I use the word osis, please note that stands for accumulations in the body. Cystinosis, ketosis. Hope you understand. Fluorosis, you are familiar with that word. When there's an accumulation in the body, we call it as osis. Acidosis, alkalosis. So here, cysteine is accumulated in the body. The reason behind is transport of cysteine is by a compound called cystinosin. That is different. Cystinuria, presence of cystine in urine. So here is a transporter. Please note this term. COAL. Cysteine, ornithine, arginine, lysine. A transporter that helps in the transport of these four amino acids. I'll repeat once again. Cysteine, ornithine, arginine, lysine. The defect in that transporter leads to a condition called as cystinuria. Then phenylketonuria, phenylalanine hydroxylase deficiency. Alkaptonuria, homogenesic acid oxidase deficiency. And one more I'll write somewhere here. It's called as Hartnup disease. Actually, a family name of an English people. And then this is because of defect in the transport of some neutral amino acid, including tryptophan. Neutral amino acid, defect in the transport of neutral amino acid, including tryptophan. That leads to a condition called as Hartnum disease. So please note all the disorders, major disorders related to amino acids. Now, about a protein, a major protein that present in the body is collagen. You know that. Predominant protein in the body is collagen. Predominant protein in the blood is albumin. You know it. 1/3 of collagen is glycine. And 1/3, and not 1/3, maybe few. Some amount is proline. So these two are the major or rich amino acid, the amino acids that are rich in collagen, proline and glycine. Okay. And about the glycine, about the collagen structure. Collagen structure, when we say each collagen molecule, triple helical structure. Okay, collagen molecule, or we call it as, the collagen molecule is also called as tropocollagen. Tropocollagen. It has a triple helical structure. So, see, I hope you can see this one, triple helical structure. And this triple helical structure, condensed form, we can say this one. So, this is one triple helical structure. See the next one. This is not just beneath that. In fact, it is little bit moved to one side. 1/4 is moved. So, this arrangement we call it as quarter staggered arrangement. So, quarter staggered. That means 1/4 is moved, quarter staggered arrangement. And this is triple helical. So the full we call it as structure of collagen, triple helical, quarter staggered arrangement. That's a structural shape, we can say about that collagen. Okay. Now a disorder related to collagen. We call it as Ehlers-Danlos syndrome, abnormal tissue fragility, hyper extensibility of the skin, increased joint movement. Joint movement we can see here, and the skin hyperextensibility is there. And this is due to the defective type 3 collagen. Here, collagen molecule, so there is a triple helical structure I said. Between them, there should be some bonds. And with the quarter staggered arrangement, that also supposed to have some bonds. For that, our lysine and glycine, lysine proline should be hydroxylated. This hydroxylation reaction, for that, we need some lysyl hydroxylase or lysyl oxidase. If there's a defect in that, so the mature collagen cannot form, that leads to a condition called as Ehlers-Danlos syndrome. One more disorder, so just note the name of this one and write the basic defect. Another defect, during the birth itself, the bonds are always fragile, osteogenesis imperfecta. Some imperfection in the genesis of osteoid. This is because some glycine amino acid glycine is replaced by cysteine. This small mutation can lead to a condition called as osteogenesis imperfecta. As I said, 1/3 of collagen is glycine. Please note that curve. And with that collagen, at least one glycine, when the single glycine is removed from the tropocollagen, that can lead to osteogenesis imperfecta. So that's all about the major questions, the major topics in the protein metabolism. I started the protein chemistry Then structure of protein, I said some points. Then I said about the amino acid classification, the compound components present the amino acids. Then about amino acid metabolism part. We classified amino acids, essential amino acids, glucogenic amino acid, ketogenic amino acid. Amino acids having maximum buffering capacity. Then groups in the amino acids. Indole group of tryptophan, imidazole group of histidine, and guanidine group in arginine, guanidine group in arginine. Name the branch and amino acid, valine, isoleucine, leucine. I said that about classification. Then some metabolism, major compound produced from each amino acid. Then major disorders related to that, and about a major protein in our body, collagen. Okay, now going to the second part of class today, carbohydrate metabolism. We'll start now. Carbohydrate chemistry and metabolism. First, I'll have some questions of carbohydrate chemistry. Which of the following is a non-reduced sugar? Glucose, maltose, lactose, sucrose. We know glucose is reducing. We are studying in the first class of biochemistry onwards. Maltose, 2 moles of glucose is reducing. Lactose, glucose plus galactose, reducing. Sucrose, glucose plus fructose, but it is non-reducing. You know why? Because the function group of glucose is linked to the function group of fructose. The function group is the one is supposed to have the reducing property. There's no free functional group. So sucrose is non-reducing. What is cane sugar? Is it glucose? No. Again, that's sucrose. What's the sugar percent in the milk? Everybody knows, lactose. What's the enzyme? What's the carbohydrate percent in the this one? Sprouted seeds? Maltose. And then one more here, fructose is there. I'll say a question on that. The sweetest sugar is fructose. So what's the fructose? Fruits is there, honey is there. So fructose is the sweetest sugar. And one more question about the fructose. The polymer of fructose. Common word for polymer, you use the word for the fructose. We call it as fructosan, but that's not the answer. Fructosan is inulin. Hope you are familiar with the term inulin that used to check the GFR. Inulin is a polymer of fructose. Glucose is the polymer. In the body, glucose is stored as glycogen. Glucose is stored as glycogen. You should know it. But in plants, the storage form is starch. The storage form of glucose in plants is starch. The animals and now body is glycogen. Okay. Which transporter is involved in the absorption of glucose? Intestinal absorption. After the food get digested, you had, think in the evening, you had some compounds which contain rice, starch. Okay. Then what will happen to the starch? What will happen to the starch? In the stomach, sorry, first in the mouth, with the help of salivary amylase, randomly breaks alpha-1,4 glycosyl linkage, produce some dextrin, partially digested compound. Then goes to stomach, we have the HCl there that inhibit the action of salivary amylase. Then intestine, it acts by pancreatic amylase. Breaks into oligosaccharides or sugars. Then we have some oligosaccharidases. Oligosaccharidases converts to glucose molecule. Now glucose, think it this way. Let this be the intestinal cell, breast border cell. This glucose to enter inside, you need a transporter here that is called as S-glut1. S-glut1. What it stands for? S for sodium, dependent, glucose, T for transporter, 1. So from the to the intestinal cell, for entry, so intestinal absorption is with the help of GLUT1, S-GLUT1. And then after entering here, to enter into our vessels, blood vessel, okay, from the enterohepatic circulation, again, there's a transporter here, that is GLUT2. GLUT2 for the transport of glucose from the intestinal cell to the blood. Okay, so my point, there are two types of glucose transporters. One is sodium dependent, other one sodium independent. So here, to the entry of glucose here, we need sodium also, so it's a symbol. But here glucose alone transport, so it's a unipod. Unipod, symbol. So this is symbol. These all are uniform. Ping pong mechanics centers. This requires energy. Requires ATP. This is passive. No need of energy. That's a major difference. Then this requires sodium. This doesn't need sodium. That's it. Okay. So S-GLUT1 is, we call it as, what is it? Okay. Secondary active transport. Secondary active transport. Okay. So this was about the glucose transport. So just not from the intestine for the absorption of glucose, you need sodium dependent glucose transporter. So whenever I teach, I say it this way. We add some amount of table salt in our all the dishes. You know why? It's not only for taste. For the absorption of glucose, for the absorption of amino acid, you need sodium. Without sodium, it cannot be absorbed. So that's the scientific reason for adding table salt in our dishes. Okay, go for next. What's the rate limiting enzyme of glycolysis? Glycolysis. As soon as the glucose enters into the cell, the main process that happens in the cell is glycolysis, where it produces energy. Okay. Okay, in that, around 10 steps are there. So we'll say one by one now. Glycolysis takes place in the cytoplasm of all cell. There are two types of glycolysis, just not all the things. Here in the PFK, phosphorus is the rate limiting enzyme. I'll come to that word. There are two types are there, aerobic and anaerobic. So in presence of oxygen, glucose converts to pyruvate. So the product is pyruvate for the aerobic glycolysis. And for the anaerobic glycolysis, the product is lactate. So make a tabular column this way. Two types of glycolysis, aerobic, anaerobic. Aerobic, the product is pyruvate, and other one, the product is lactate. How much energy? That's the next question. In presence of oxygen and mitochondria, I should say it that way, conditional. In presence of oxygen and mitochondria, you get 7 ATPs here and 2 ATPs there. Because there's no mitochondria, that 5 cannot be produced from by the process of oxygen phosphorylation will not be there. So 7 and 2. Types we said the product, energetics we said. Aerobic we get 7. And then what's the next site? takes place in all the cells in the cytoplasm. And what are the significance of glycolysis? This is the only source of energy for RBC. Please note, that's one important factor. Our RBC depends only on not just glucose, we can say more specifically, only on glycolysis for energy. Because all of the process energy production mainly takes place in the mitochondria. Mitochondria is the powerhouse, but RBC doesn't have a mitochondria. Glycolysis takes place in the cytoplasm. Two subcellular phosphorylation takes place. So RBC, the energy from glycolysis. And what is the other importance of that? There is a shunt pathway, bypass pathway for glycolysis. We call it as 2,3-BPG shunt. A compound is produced 2,3-bisphosphoglycerate. Compound is 2,3-bisphosphoglycerate. And this compound can reduce the affinity for hemoglobin. Hemoglobin has a strong affinity for oxygen, very strong association with the oxygen. If that 2,3-BPG is there, that can reduce affinity for oxygen. So it can easily leave out or empty, or it can give out the oxygen. So oxyhemoglobin converted to deoxyhemoglobin in some cells we need, or in presence of 2,3-BPG, it can release out more oxygen. It can unload more oxygen. That's very important. That pathway is called as 2,3-BPG shunt or Rappaport lubricant cycle. I'll come to that word later. One more. In the step, there is a step called as enzyme where we use is enolase. That is inhibited by fluoride. What is the importance of that? See, early morning, think it this way. Early morning sample, we take the test tube or blood sample. It contains RBC cells. This RBC cell will utilize the glucose inside. So keeping the glucose level decreases. You want to check the glucose level. You took the sample at 6 o'clock, maybe for the mass of fasting blood sugar. But you are taking it to the lab only by 8 or 9. What happened? Every hour, around 10 milligram decreases. So, to inhibit that process, inhibit the glucose uptake by RBC, we add some amount of fluoride. Why fluoride? That inhibits the enolase enzyme in the glycolysis. Thereby, the glycolysis process will be inhibited. So, RBC will not be able to utilize glucose. That's the point. There are three enzymes, rate-limiting enzyme. We call it as HK, PFK, PK. Hexokinase, phosphokinase, and pyruvate kinase. Out of which, this is the most important, PFK. That's why I use the word, the major rate-limiting enzyme of glycolysis is phosphofructokinase, PFK. Please note the enzyme name, PFK. Three enzymes are there, HK, PFK, PK. Three irreversible steps for that three enzymes. Okay. This is about our cycle, our shunt pathway. In glucose, there's a conversion from 1,3-bisphosphoglycerate to 3-phosphoglycerate. In RBC alone, please note, in RBC alone, this 1,3-bisphosphoglycerate will convert to a compound 2,3-bisphosphoglycerate. Then 2 again back to 3-phosphoglycerate. This pathway, bypass of glycolysis, we call it as 2,3-BPG shunt, BPG shunt, bisphosphoglycerate shunt pathway. And or other words, it's also called as Rappaport lubricant cycle. Please note the word Rappaport lubricant cycle. That's a shunt pathway of glycolysis. Shunt pathway of glycolysis. Where this shunt pathway takes place only in RBC to produce 2,3-BPG. That is to reduce the affinity of hemoglobin for oxygen. So the RBC can easily download, or hemoglobin can easily download the oxygen. Next, inhibition of glycolysis by oxygen is known as a question. Just identify this program or uh, this was identified by a person called as Pasteur. So this is called as Pasteur effect. You know why? In presence of oxygen, a lot of ATP will be produced. ATP is the end product, so it will allosterically inhibit the glycolysis. So that's why inhibition of glycolysis by in presence of oxygen, is known as Pasteur effect. And we have one more effect, Warburg effect, just opposite of that. But in cancer cell, even if you are having oxygen, there will be glycolysis, then the more anaerobic glycolysis. That's called as Warburg effect. Cancer cell prefers anaerobic glycolysis and produce lactic acid even in the presence of oxygen. This is called as Warburg effect. Because acidic nature is very important for the cancer cell to grow and for the metastasis. So it depends on that. Okay, going for next. After glycolysis, what happened to the pyruvate? Pyruvate and alpha ketoglutarate, the same pathway, it's a multi-enzyme. Both are contain 3 enzymes and 5 coenzymes. The step is called as, usually the pyruvate will convert to lactate in anaerobic condition. In aerobic condition, in the mitochondria, the pyruvate will convert to acetyl-CoA. It's pyruvate dehydrogenase complex. Example for oxidative decarboxylation. So there are 3 oxidative decarboxylation steps in our body. One is this, pyruvate to acetyl-CoA. Second is a conversion of alpha ketoglutarate to succinyl-CoA. Third, I have already said that term, our alpha keto acid of branch chain amino acid. Just now I said. To its corresponding acyl thioesters. Acyl thioesters. These 3 are example for oxidative, see, check the word, oxidative decarboxylation. And if this step, when it inhibits, we call it as MSUD. Hope you remember that term. Okay, in all these things, we have enzyme. Here it is pyruvate dehydrogenase complex, there it is alpha ketoglutarate dehydrogenase complex, here it is alpha branch and alpha ketoacid dehydrogenase complex. All 3 contain 3 enzymes and 5 coenzymes. That's the point. These all contain 3 enzymes and 5 coenzymes. So it's a multi-enzyme. So this word contains 3 enzymes. Pyruvate dehydrogenase, dihydrolipoid transactylase, dihydrolipoid dehydrogenase. So in all 3, the second and third will be same. The first alone, the first pyruvate to acetyl-CoA, pyruvate dehydrogenase. In alpha ketoglutarate succinyl-CoA, alpha ketoglutarate dehydrogenase. In branch chain alpha ketoacid to acetyl-CoA esters, branch chain alpha ketoacid dehydrogenase. So there are 3 enzymes in that complex and 5 coenzymes or 5 vitamins. Coenzyme A, niacin, lipoic acid, thymine, tryptophan. Hope you have taken this is an important question. At least a coenzyme of vitamin percent. Please take a screenshot of this one. Okay, going for next question. It's all about all of the following are the substrate or the sources of gluconeogenesis, except, of course, what you should know, what is gluconeogenesis? Genesis means synthesis. You know the word, synthesis. Synthesis of glucose. By the word neo, new. From means it's not from carbohydrate, from somebody else. That means synthesis of glucose from non-carbohydrate sources. We call it as gluconeogenesis. What are the non-carbohydrate sources? Our glucogenic amino acid is one source. The first and foremost, most important, lactate. Second, glucogenic amino acid. Third, odd chain fatty acid alone, not all fatty acid, odd chain fatty acid. Fourth one, glycerol. Now check the point. We have alanine. It can produce glucose, glucogenic only. Only ketogenic is, as I said about what? Purely ketogenic is lucid. We said that. Oleic acid is not odd chain, so it cannot. Glycerol produce. Tryptophan is also again glucogenic amino acid. So the answer here is oleic acid. Only odd chain fatty acid can produce glucose. Okay, what? So I said gluconeogenesis. Site is in mainly liver, 90% liver and 10% renal medulla. Sources we said, site we said now. Energetics. In glycolysis, around 7 ATPs are produced. But when you go back, totally around 6 are utilized. Anyway, that's also important. Significance, whenever we have hypoglycemia, whenever we are on starvation, prolonged fasting, this happens for the RBC, the bus, or the only source is glycolysis. So always glucose support is essential. Glucose supply is essential for RBC. Brain also likes to have the glucose, where it cannot utilize fatty acid. So brain cells, renal medulla, some nerve tissues, nerve cells, they all utilize glucose. This is what glucose for energy. So whenever we are on starvation, these cells, we provide glucose through gluconeogenesis. Understand that way. Now comes next topic, TCA cycle. What is TCA cycle? Okay, here the question, there are two types of ATP synthesis. Two types of ATP synthesis. One, when a substrate is converted to product, at that time itself, ADP converts to ATP. This is called as substrate level phosphorylation. In our TCA cycle, there is only one step. This is about thiokinase, succinate to succinyl-CoA. Glycolysis, we have two. Glycolysis, we have two substrate level phosphorylation. Okay. We'll go through the cycle now. Don't worry. And order. We'll come back in which order it goes. This first one only citrate to oxalate, alpha glutamate. We'll just check it out now. This is the Krebs cycle. Acetyl-CoA from outside enter by combined with oxaloacetate. So this is one irreversible step. Irreversible step number one. Then second is citrate, water addition and then removal. We have a acid-saccharide in between anyway. Major compound isocitrate. Isocitrate, we have an intermediate compound oxide or succinate. Leave that. Converts to alpha glutamate. At that time, see, NADH plus H is produced. This one, from this one, electrons are removed. Removal is called as oxidation. It goes to a protein, set of protein, and combined with oxygen to form water. At that time, ATP is produced. So during oxidation, ATP is produced. So the word oxidative phosphorylation. So there are two types of phosphorylation. Earlier starting, one is called as, I've written that, no? One is called as substrate level phosphorylation. This one is substrate level phosphorylation. A substrate converts to a product. At that time, it will produce. Second one, as I said now, from the NADH, we call it as oxidative decarboxylation. Sorry, oxidative phosphorylation. So one is substrate level phosphorylation, other one is oxidative phosphorylation. Here, NADH undergo oxidative phosphorylation, produce 2.5 ATP. Then alpha-ketoglutarate converts succinyl-CoA. Again, another reversible reaction. This is also another reversible reaction. Second, three. There are three irreversible reactions in TCA cycle. Here also one NADH is produced. 2.5 ATP. 2.5 ATP. Then here one GDP is converted to GDP. That's what I said, succinyl-CoA succinate. Here, example for substrate phosphorylation, equivalent to 1 ATP. Then succinate fumarate, one FADH is produced, it produces 2.5 ATP. Sorry, 1.5. From FADH to 1.5. So, and then last malate oxalate, 1 NADH. So 2.5 ATP. So totally 2.5, 2.5, 2.5, 7.5. Then 1.5 here, 9 plus 1 here. So totally 10 ATPs. TCA cycle produced 10 ATPs. TCA, T, 10. Study like that. This one. How many? So next question. Okay, so for this cycle, 10 ATPs and study the steps. Citrate, isocitrate, alpha-ketoglutarate, succinyl-CoA, succinate, fumarate, malate, oxalate. Next question, how many ATPs are produced in the complete oxidation of glucose? Glucose converts to pyruvate. There we get 7 ATP. Then pyruvate to acetyl-CoA. 2 pyruvate are there, 2 acetyl-CoA, so 2 NADH. 2. So 5 ATP is there. Then this acetyl-CoA enters into the cycle. When one acetyl-CoA enters, 10 ATP. 2 acetyl-CoA, 20. So 5, 20. Totally how much? 32 ATPs are produced from on breakdown of one glucose molecule. Okay, go for next question. Is about glycogen. Glycogen, what is glycogen? Storage form of glucose. Okay, and there are two enzymes mainly for glycogen, rate-limiting enzyme. I write the name here. For synthesis, we need an enzyme. The rate-limiting enzyme is glycogen synthase. For breakdown, and another enzyme is glycogen phosphorylase. Phosphorylase 2 enzyme. Number of other enzymes are there. Now just go through. Here I'll just show you later. MCQ is a disease related to glycogen storage problem, storage disorder. McArdle is because of myophosphorylase enzyme deficiency. And the glycogen storage disease some persons will pronounce it as Giacch's disease or Giacch's disease because it's a German scientist. Von Giacch's disease. There you can see hyperuricemia. It is type 1. So these are the storage disorder related to glycogen. Von Giacch's disease is glucose 6-phosphatase. Pompe's disease or Pompe disease, lysosomal maltase. Cory's disease, CD, debranching. Anderson disease, branching enzyme. McArdle disease, muscle phosphorylase. Her's disease, liver phosphorylase. Torrey's disease, muscle PFK. PFK means phosphatidylase. Eighth and we don't have the much name for it. One is liver phosphorus kinase, other one muscle phosphorus kinase. So one question can come from here by just asking you the enzyme deficiency of each. By giving the name of the defect, can ask you the reason behind. This is about the glycogen storage disease. And I'll say about one glycogen storage disease or von Wieker's disease. What happened there is glucose 6-phosphate deficiency. Usually the glycogen is mainly you convert to glucose when you are fasting. Here glycogen is not able to break. So what will happen? This leads to fasting hypoglycemia. Understand in this way. In the body, from the liver, whenever you are on starvation or fasting, after 3 hours of fasting, the glycogen breaks to, first converts to glucose 1-phosphate, then glucose 6-phosphate. After this part in muscles and liver is same. But in the liver, this glucose 6-phosphate with the help of enzyme glucose 6-phosphatase converts to glucose. This happens only in liver. Enzyme is glucose 6-phosphatase. And that enzyme deficiency, what happened? Glucose 6-phosphatase is not converted to glucose, so fasting hypoglycemia. Then what happened? Hyperlipidemia. Because there's more lipolysis from the adipose tissue, it goes to comes to circulation. Third, lactic acidosis. Glucose is not synthesized from lactate, so there will be the same enzyme, glucose oxidase is there in gluconeogenesis. Lactic acidosis. 4, this is what? This glucose 6-phosphate, instead of going to convert to glucose, this enters into HMB pathway, will teach that it helps in the production of two compounds, NADPH and pentose. From the pentose, you get uric acid. So, in the absence of glucose 6-phosphatase, I just show that glucose 6-phosphate enters into HMB pathway. HMB pathway will produce ribose. Ribose produce nucleotides. Nucleotides are converted back to uric acid. Or uric acid is the end product of purine or pyrimidine nucleotide metabolism. So von Wieker's disease, hyperuricemia, fasting hypoglycemia. Please note that. Glycogen storage disease 1. Glucose 6-phosphate. So this is what? Von Wieker's disease. Can pronounce it that way too. Blood glucose level cannot be augmented by the mobilization of muscle glycogen due to lack of water. Just now I said. Glycogen converts to glucose 1-phosphate first, then to glucose 6-phosphate, then to glucose. Then only the glucose only can come out of the cell to the blood. For the conversion of glucose to glucose, liver we have an enzyme called as glucose 6-phosphatase. But in this enzyme is absent in muscle. So glucose 6-phosphate is not converted to glucose, so it cannot go out to the blood. That's why blood glucose level cannot be augmented, cannot be supported by the mobilization of muscle glycogen, but liver glycogen can because of the absence of enzyme, glucose 6-phosphatase. It's present in the liver. But in the liver, if it is absent, that leads to a condition called as von Wieker's disease. Okay. Now, next pathway of carbohydrate metabolism, HMB pathway. HMB pathway, first understand that here, one step in that is trans-ketolase that needs thymine. So TPP is the first B1 active form of thymine pyrophosphate. For the trans-ketolase active, there are 2 steps in the HMB pathway. There with the action of where trans-ketolase is acted, and that for the action of transketolase, you need TP. Okay. HMB shunt pathway, it's a pathway. Usually the glucose pathway is for energy. But this pathway is not for energy. It is for two compounds, NADPH and pentose. Energy is zero. HMB pathway is not for energy. HMB pathway is to produce NADPH and pentose. With NADPH, you can synthesize lipid. You can synthesize glutathione in the reduced form. You cannot synthesize glutathione, but you can keep the glutathione in the reduced form. You can remove the free radicals. You can synthesize nucleic acid. Okay, so these are some uses of NADPH. Pentose is for nucleic acid synthesis. This is what shunt means: divert, switch or bypass. Usually glucose is converted for energy purpose. But now here, glucose is converted to pentose phosphate. And with the help of and during which NADPH is produced. So both NADPH and pentoses are the products of HMB pathway. No energy. That's the point. Okay, next. So HMB pathway, this is important point. And the rate limiting enzyme is glucose 6-phosphate dehydrogenase. Please note, G6PD is the rate limiting enzyme. Please study all the rate limiting enzymes of each pathway of glucose. In glycolysis, I've already told you HK, PFK, PK. Glycogen metabolism for synthesis, synthase, and breakdown phosphorylase. HMB pathway, the rate limiting enzyme, glucose 6-phosphate dehydrogenase. In gluconeogenesis, we have 4 enzymes. Glucose 6-phosphatase, fructose 1,6-bisphosphatase, pyruvate carboxylase, phosphoryl pyruvate carboxykinase. Okay, 4 enzymes. HMB pathway takes place in all the sites where it produces what we say lipids, because it's involved in lipid biosynthesis. Okay, and which all tissues can want to scavenge the free radicals. There also we need the HMB pathway will take place. NADPH is produced. NADPH will helps to maintain the level of antioxidants. Okay, coming back now, that's all about the HMB pathway. Next comes about galactose metabolism. There's a disorder called as galactosemia. Enzyme deficiency, galactose 1-phosphate uridyl transferase. A kid with galactosemia will be having brain damage, cataract, jaundice, enlarged liver, kidney damage. Whenever he have the milk, he have the problem. And here, the most important, see the word cataract. We always see cataract only in aged patients. But here, what happened? Galactose is converted to a compound galactitol. Galactitol. This galactitol accumulation can lead to cataract. That's one question. In diabetes mellitus, the cataract is because of a compound called as sorbitol. In diabetes mellitus. But in galactosemia is because of accumulation of compound galactitol. Or one more word is there, dulcitol. Dulcitol accumulation can lead to cataract. Enzyme deficiency, galactose 1-phosphate uridyl transferase. I'll show the this one schematic representation about the galactose metabolism. Galactose first convert to galactose 1-phosphate. That converts to UDP galactose. There comes the enzyme. There comes our enzyme, galactose 1-phosphate uridyl transferase. And the absence, this will not take place. So this accumulates, that converts to galactitol or dulcitol. that leads to cataract. Please note. Okay, maybe the last question. How much is the real threshold of glucose? Glucose, the normal FB, SPB level, fasting blood sugar level. Nowadays, it is called as 70 to 100 is the FBS. PPBS up to 140. Should be less than 140. Should be less than, sorry. Less than 140 milligram per deciliter. That's what remember stage. Now, usually, in the patient, in any subject, urine sugar will be absent if it is a normal blood sugar level. If the blood sugar level goes more than 180, then only the sugar will be present, glucose will be present in the urine, will be excreted through the urine. This level we call it as renal threshold. So what is the renal threshold? Capacity of the kidney up to which it can hold the glucose excretion is called as renal threshold. It is 180. Please note. And some patients, what happened? The renal threshold is capacity decreases. Then glucose will be present in the urine, glucosuria. Because of kidney's problem, that's called as renal glucosuria or glycosuria. Both ways. Renal glycosuria or this is because of decreased renal threshold. So study these terms, the normal value of fasting blood sugar, reference site of PBBS, renal threshold, and the word renal glycosuria. Yes, I'll have one more question to open the door into the lipids. In prolonged starvation, what's the main energy source of the brain? Glucose cannot be utilized. You're on starvation. Fat acid cannot enter because fat acid always combined with albumin. So it cannot cross the blood-brain barrier because they can transported by the albumin. Then at that time, our source of energy for the brain is ketone bodies. For all cells, we will study that tomorrow. Comes in lipid metabolism. Just understand that for the brain, the main source of energy is glucose, but in its absence, ketone body is the main source of energy. So I will stop here for today. If there are any doubts, you can just write down in your chat box. We will discuss that later, maybe in the next class. So shall I stop here? So today I was teaching you, I was just going through the major questions of protein metabolism and carbohydrate metabolism. The next class we will study about lipid metabolism. Thank you.